Hi there, hope you are fine. This is Mr. Philip Koma, the excellent biology tutor. We're looking at animal nutrition, and this is part 2. Without wasting much of your time, let us begin. So what are the objectives for this tutorial video? What are the objectives for this tutorial video? We're going to look at digestion in the stomach. We're going to look at digestion in the duodenum. We're going to look at digestion in the jejunum. And finally, we're going to look at the ilium and we're going to look at absorption. So in the previous video, we looked at digestion in the mouth, and we also looked at peristalsis and the movement of food along the alimentary canal or the esophagus. So I hope you still remember what we did in the previous video. So yeah, let's look at digestion in the stomach. All right. So the stomach is an elastic bag with a muscular wall and a glandular lining. Okay, simply an elastic bag. It's a bag which stores food temporarily. Okay, it's a bag, it's an elastic bag. The entrance of the stomach is guarded by cardiac sphincter. These are the muscles that are found at the entrance. Okay, they are rings of muscles found at the entrance of the stomach. And what do? They guard. Okay, and then the exit is also guarded by muscles, and the name of these muscles are the pyloric sphincter. The ones at the entrance are the cardiac sphincter, then the ones at the exit are the pyloric sphincter. Now, the following events take place in the stomach. So, remember, when you put food into your mouth, the first thing that you start doing is to chew it, isn't it? And then that food is going to be mixed with saliva, and we know what saliva contains. You remember that, right? Yes. So in the stomach, immediately the food reaches the stomach. Immediately it reaches the stomach, there is secretion of gastric juice. Gastric juice is going to be secreted. Now, gastric juice is a mixture of pepsin, renin, hydrochloric acid, and mucus. These are the contents of gastric juice. Pepsin and renin are enzymes. All right, these are enzymes. Now, what's the function of pepsin? Pepsin simply breaks down proteins to form peptides. Remember that enzymes are the ones that are responsible for chemical digestion, right? Yes. So pepsin breaks down what? Proteins to form peptides. So it is the pepsin that breaks down any protein that is in your food. They break it down to form peptides. Renin, on the other hand, coagulates milk by converting the soluble protein caseinogen into an insoluble form called casein. So for renin, it coagulates. To coagulate simply means to make something to become semi-solid. Okay, they coagulate milk by converting the soluble protein, which is the caseinogen, into an insoluble form called casein. So the soluble protein is known as what? Caseinogen. The insoluble one, it is known as what? Casein. So renin simply coagulates milk. Okay. Why is coagulation important? Because this delays the passage of milk to the duodenum. So it gives it more time for pepsin to digest proteins. So that milk will be made it will be converted into a semi-solid substance to just give it more time for pepsin to break down the proteins that is in milk. All right, hope you are clear. So both pepsin and renin are secreted in inactive forms called pepsinogen and prorenin. So when pepsin is secreted and renin is secreted, they are inactive. They are not active. They are what? They are inactive. Okay? They are inactive. When pepsin and renin are secreted, they are not active. They have to be activated. So as they are inactive, their names are pepsinogen and prorenin. Okay. So that's the function of pepsin and renin. Pepsin simply breaks down proteins to form peptides. Renin coagulates milk. And I explained why coagulation is important. Now, the other content of gastric juice is hydrochloric acid. Okay. what is the function of hydrochloric acid? Hydrochloric acid activates uh, uh, the enzymes, which is pepsin and renin. Remember, when they are secreted, they are inactive. They are now activated by what? Hydrochloric acid. So hydrochloric acid activates them into active enzymes and sets an acidic pH which is optimum. So the stomach is an acidic medium. The mouth is what? Slightly alkaline. I hope you remember this. The mouth is slightly alkaline. But for the stomach, it is an acidic medium because of hydrochloric acid. So hydrochloric acid simply activates the enzymes, which are renin and pepsin, okay? And then it also sets up an acidic environment, okay? An acidic pH, which is optimum. So when we're talking about optimum pH, we're simply talking about the medium which is going to be suitable, or which is going to be favorable for the enzymes in the stomach to act at their best, to perform at their best. It's the same with optimum temperature. At optimum temperature, enzymes will work at their best. So it's equal even with pH, okay? So if the pH is optimum, meaning at that pH value, the enzymes will work at their best. So hydrochloric acid activates the enzymes, and it also sets an acidic pH which is optimum. That's not the only function of hydrochloric acid. The other one is that it also kills bacteria which may come with the food, and it hydrolyzes sucrose to glucose and fructose. It does what? Hydrolyzes sucrose to glucose and fructose. We've got another content of gastric juice, which is mucus. And mucus simply protects the lining of the stomach against the acid and pepsin. So mucus simply protects the lining of the stomach against the acid and pepsin. So these are the contents of gastric juice and their functions. So immediately food reaches the stomach, this juice known as gastric juice is secreted. All right. Now, churning is another process which takes place in the stomach. What is churning or canning? This is the mixture of food by rhythmic contraction of the muscles in the wall of the stomach to form a paste called chyme. Remember, in the mouth, food is rolled by the what? By the action of the tongue and the palate to form a bolus. And that bolus is the form of food, the new form of food which we now swallow, right? In this case, it is canning. Canning is going to take place. Canning is the mixture of food by rhythmic contraction of muscles in the wall of the stomach to form a what? To form a paste. And this paste is now what? Chyme. So from bolus to what? To chyme. Alright. So temporal storage of food. The stomach stores food temporarily. It stores what? Food temporarily. So liquids can stay in the stomach for about 30 minutes. Okay? They can stay in the stomach for about 30 minutes. Sorry about that. So liquids in the stomach can stay about 30 minutes. And then carbohydrates are kept for about an hour. Proteins and lipids stay up to 2 hours. So liquids can stay in the stomach for about how many minutes? 30 minutes. Carbohydrates are kept for about an hour, and then proteins and lipids stay up to 2 hours. Alright, my students, let us continue. Now, once chyme has been formed from the stomach, it will now go into the duodenum. Okay, it will now go into where? The duodenum. So, the duodenum receives digestive juice from the liver and the pancreas. The duodenum receives digestive juices from the liver and the pancreas. So in the duodenum, it is the digestive juice which is secreted by the liver and the pancreas. So in the stomach, it is gastric juice. In the mouth, it is saliva. In the duodenum, it is what? Digestive juice. Okay, I repeat. Stomach, it's gastric juice. In the mouth, it is saliva. In the duodenum, it is digestive juice. Now, digestive juice is secreted by what? By the liver and the pancreas. The liver secretes bile, which is a digestive juice. This bile is temporarily stored in a gallbladder. I don't know if you still remember. I hope you still remember the diagram that I showed you in part 1. There was that diagram which was showing the alimentary canal in a man. That diagram, when you look at it, you're going to see each and every part that has been explained here. Okay, so the bile, which is secreted by the liver, it is stored by the gallbladder, and then the bile is going to be carried to the duodenum by the bile duct. Now, bile is a digestive juice. It's a juice, and this juice contains what? Contains sodium hydrogen carbonate. It contains bile salts and bile pigments. The bile, it is a digestive juice which is secreted by the liver. And this bile contains what? It contains sodium hydrogen carbonate, it contains bile salts, and it contains bile pigments. Now, what's the function of each component that is found in bile? Sodium hydrogen carbonate simply neutralizes the acidic chyme and then sets an optimum alkaline pH for the enzymes of the duodenum. So remember the chyme which was formed in the stomach? Yes, that chyme is acidic, and the duodenum is not an acidic environment. It is an alkaline environment. So it is the duty of sodium hydrogen carbonate to neutralize the acidic chyme and to make it alkaline. Okay, it's the duty of the sodium hydrogen carbonate to do that. Now, after neutralizing the chyme, it also sets up an alkaline pH for the enzymes that are found in the duodenum. Remember, pH, when we say optimum pH, it is the pH value at which an enzyme will work at its best. And these pH values, they range from 1 to 14. I hope you know we did enzymes. This is explained under enzymes. The pH value ranges from 1 to 14. So when I say optimum pH, it is simply a pH value at which an enzyme will work at its best. So in the duodenum, the pH is alkaline. In the stomach, the pH is acidic. Okay, so sodium hydrogen carbonate will set up the optimum alkaline pH for the enzymes in the duodenum. Alright, so that's the function of sodium hydrogen carbonate. The bile salts, on the other hand, the bile salts emulsify fats, thereby increasing the surface area for the action of lipase. They do what? The bile salts emulsify fats. What does that mean, to emulsify? They break down fats. Okay, so they break down what? Fats. And then why do they do this? To increase the surface area for the action of lipase. Lipase is an enzyme, my students. Lipase is an enzyme. So now, what is emulsification? Because we are saying that bile emulsify. So emulsification is the breakdown of large drops of fats into smaller droplets. So they're just going to break them down. They're going to break down fats. Bile salts will break down fats into smaller droplets to increase the surface area for the action of this enzyme known as lipase. So that's the function of bile salts. We've got another content of a bile, which is bile pigment. Bile pigments have no digestive function. They have no digestive function, okay? But they add color to feces. That's what bile pigments do. They simply add color to the feces. The pancreas is another organ which secretes digestive juice. But before we even look at the pancreas, I want us to go a bit back to say these things again, because we learn through repetition. So I said that digestive juices, these are juices that are secreted by the liver and the pancreas. So the liver secretes a digestive juice known as bile, and bile contains sodium hydrogen carbonate, bile salts, and bile pigments. The function of sodium hydrogen carbonate is to neutralize the acidic chyme and set an optimum alkaline pH for the enzymes of the duodenum. Then the function of the bile salts is to emulsify, meaning to break down fats into small droplets, to increase the surface area for the action of lipase, which is an enzyme found in the duodenum. And we know emulsification is the breakdown of large drops of fats into smaller droplets. And then bile pigment only adds color to feces. That's the function. All right, we are clear on that one, right? Let's look at another digestive juice which is secreted in the duodenum. So the pancreas secretes pancreatic juice, which contains sodium hydrogen carbonate, trypsin, lipase, and pancreatic amylase. These are the things that are contained in the pancreatic juice. Under the endocrine system, we are going to learn that the pancreas also secretes, it's also made up of cells known as the islets of Langerhans that are responsible for the secretion of hormones. So the pancreas is known to be an endocrine gland and exocrine gland. It is an exocrine gland in the digestive system, and these are the things that it secretes in the digestive system. It secretes what? Pancreatic juice, which contains sodium hydrogen carbonate, trypsin, lipase, and pancreatic amylase. These are the three things that are found in the pancreatic juice. Now, sodium hydrogen carbonate neutralizes the acidic chyme, as we know, and it sets an optimum alkaline pH for the enzymes of the duodenum. Trypsin, what does trypsin do? Trypsin breaks down proteins. Okay, trypsin does what? Breaks down proteins to form peptides, just like pepsin in the stomach. So here, if there was any protein that wasn't digested in the stomach, it will also be digested here. So trypsin breaks down what? Proteins to form peptides. Excuse me. Then lipase breaks down fat molecules to fatty acid and glycerol. So when lipase breaks down fat molecules, what is going to be formed? Fatty acids and glycerol. All right. The pancreatic amylase breaks down starch to form maltose. In the mouth, we've got the salivary amylase, right, which breaks down starch, cooked starch, to maltose. And remember, we said that not all starch is digested in the mouth, because food will only stay for a few minutes in the mouth and then you swallow. So other starch which are contained in the food will be broken down here in the duodenum by the pancreatic amylase, because the function of the pancreatic amylase is to break down starch to form maltose. Are we clear, my excellent students? All right. Let us move on. Digestion in the jejunum. So in the jejunum, there's another juice known as the intestinal juice. So in the mouth, saliva; in the stomach, gastric juice; in the duodenum, digestive juice; in the jejunum, intestinal juice, which is also known as succus entericus. So this juice contains lactase, maltase, sucrase, and peptides. This juice contains what? Lactase, maltase, sucrase, and peptides. Lactase breaks down lactose to glucose and galactose. Now, I want you to follow me very well. Under nutrients, glucose will combine with galactose to form lactose. Pause there for a minute and think. Pause there for a minute and think. Glucose and galactose, these are simple sugars, my students. They are simple sugars. They are going to combine, they will add up to form what? Lactose. So lactose is made up of glucose and galactose. Are you following? Yes. But this lactose, which is made up of glucose and galactose, when lactase breaks it down, it simply means it is going to break it down into its original component, into the components that made it. So lactase breaks down lactose to form glucose and galactose, which are the components that make up lactose. Yes. Maltase breaks down maltose to glucose. Glucose plus glucose will form maltose. Glucose plus glucose will form what? Maltose. So when maltase breaks down maltose, it is going to take it back to its original components, which is what? Glucose. Sucrase breaks down sucrose to glucose. So sucrase will break down what? Sucrose. So sucrose is is made up of glucose and fructose. So glucose and fructose are going to form what? Sucrose. So when sucrase breaks sucrose down, what will be formed? Glucose and fructose, which are the original components of sucrose. Peptidase breaks down peptides to amino acids. Under ecology, there is the process of deamination, okay, which is explained even there. So peptidase breaks down what? Peptides to form amino acids, which are the building blocks of proteins. Are you following? So proteins were digested to form what? Peptidase or peptides. Yeah? Yes, peptides. In the stomach, proteins were digested to form what? Peptides by pepsin. In the duodenum, proteins were also digested again. In this case, the peptides which are formed when proteins are digested will be broken down again by peptidase to form amino acids. And then digestion is completed in the jejunum. This is what happens in the jejunum, my excellent students. So it's a matter of you watching this video over and over again, because that's a lot of information there. So the ilium carries out absorption. Okay, the ilium now carries out the process of absorption. Absorption is the next stage. After digestion, there's absorption, assimilation. There's ingestion, digestion, absorption, assimilation, egestion. So we are now done with digestion. We're now looking at absorption. So this process of absorption is carried out by what? The ilium. So the ilium carries out absorption of digestive end products and is adapted for this function in the following ways. So the ilium, which you're going to later on see, it is the one which carries out absorption. What gives it the qualification? What makes it possible for it to perform its function? The first characteristic or adaptation of the ilium is that it is very long, thereby providing a large surface area for absorption. The ilium is very long, which gives it the ability to have that large surface area for absorption. It has a thin epithelium for more efficient diffusion of food. We're going to look at the diagram, so don't worry. Epithelium it's a layer, okay? So it has a thin epithelium, which is a layer. And since it's thin, diffusion is going to be efficient because it won't have to take time for things to move between the two regions, okay? So it has a thin epithelium for more efficient diffusion of food. It has finger-like projections called villi, singular villus, and microvilli, which further increases the surface area for absorption. So it has got finger-like projections called what? Villi, singular villus. And these, they further increase the surface area for absorption. Then each villus or villi has a network of capillaries for absorption and transportation of monosaccharides and amino acids. So when you hear the term capillaries, blood should come into your mind, because then the food is absorbed and then it is taken into the blood, okay? And these capillaries are the ones that are linking blood to the villus. Each villus has a lacteal which absorbs and transports fatty acids and glycerol. The lacteal does what? Absorbs and transports what? Fatty acids and glycerol. Alright, this is the diagram now. The epithelium, remember the epithelium, which we said it is long. Okay, so this is the epithelium, as you can see, the epithelium. So this is the epithelium, my students. This is the epithelium. And then we've got the lacteal. What did we say the lacteal does? You've seen this is the lacteal, lymphatic vessel. This here is the lacteal. What did we say the lacteal does? Each villus has a lacteal which absorbs and transports what? Fatty acids and glycerol. So fatty acids and glycerol are transported by the lacteal. Then this here is the capillary, where blood is flowing. Okay, this is a capillary where blood is flowing. This is the arterial, and then we've got the venule. Yeah. So this is the finger-like projection. You've seen this thing. If you look at this, these are now these are finger-like projections. Now these are finger-like projections which are leading to this. Okay, these are finger-like projections. All these are finger-like projections. So taking you back, the ilium. The ilium is a tube. It's like a tube. Please watch the first part and look at that diagram for the alimentary canal in man. You find that the ilium is the tube, which is this. This is the tube, okay, in a tube, ilium. And this tube has got finger-like projections because the ilium, it is the one which is responsible for absorption. So it has got finger-like projections which are known as what? Villi. Dear to finger-like projections. So to zoom this thing, it is here. So when your food is passing through this tube, the nutrients are now absorbed. The food is now absorbed. Okay. And how is it absorbed? Because the layer, you've seen this layer. Let me try to zoom this. So the outer layer, it is the one we call the epithelium, okay, which is thin to increase what? Diffusion. So this is the layer. This is the layer to increase what? Diffusion. And then inside the villi, we're going to find these are blood vessels. So there's going to be diffusion of food from this environment going into the villus, and then since the layer is thin, food will be able to diffuse at a faster rate from this side. Imagine this hand is food, so it's going to move from this side to diffuse going into this, and then the blood capillary will take it up. Okay, yes. So yeah, this is what you need to know about the ilium and the villus. The villus is the one which increases the surface area for diffusion, because they are a lot. As you can see, they are a lot that increases the surface area for diffusion. All right, so yeah, surface area for diffusion or absorption. All right, my excellent biology students, that's the end for this part. Watch out for the next part, which is part 3. This has been Mr. Philip Kauma, the excellent biology tutor. Stay blessed.